Good morning or good afternoon. My name is James Branley. I'm an infectious diseases physician and clinical microbiologist. And I would like to talk to you today about molecular medicine, particularly with reference to the diagnosis of infectious diseases. The scope of this talk is to talk about, firstly, some definitions, then a brief discussion about host-pathogen interactions, moving on to discussing the diagnosis of infection and detection of resistance genes, and then finishing with some discussion about whole genome sequencing and typing for epidemiology. So I find the terminology in this area somewhat daunting. And I just wanted to run through a few terms just so that the audience is aware that there are some different words that are thrown around in this space that need explaining. I've used the term molecular medicine, which I guess is an umbrella term covering genetics, covering genomics, and covering the protein expression of genes and the clinical effect of that. I guess molecular medicine is a very broad term. Genetics, on the other hand, refers to the study of genes themselves. Genomics refers to the study of whole genomes. And proteomics refers to the protein equivalent of that, the whole protein expression of an organism. In clinical medicine, we're now starting to talk about precision medicine and individual therapy, which is really the application of these sciences to the individual patient in front of us. Just briefly, a couple of words about PCR and NAAT testing. The broad term, once again, is nucleic acid amplification techniques, NAAT testing. Whereas PCR is a specific NAAT, it's a specific amplification technique and the most commonly used, so we often use these terms interchangeably. So when we look at nucleic acid amplification techniques, PCR is the most common and we're very familiar with that, but there are other techniques. There is a technique that's not used much anymore called ligase chain reaction, which again is a chain reaction amplification technique. There's also loop amplification, which is isothermal. That's one technique that doesn't involve cycling the temperature in order to get amplification of the DNA. There are other techniques as well that I won't go into in the scope of this talk. Just a quick refresher, and I'm sure many of the audience understand PCR very, very well. But the basic steps in PCR involve extraction of the DNA or the RNA from the specimen, mixing that with a mix of base pairs and primers, and then naturing and denaturing that specimen with cycling of temperatures. Now we do know that as a product is generated, it can be detected in numerous different ways, and there's been a lot of recent advances in quite clever product detection. Along with that product detection, the number of cycles involved in identifying that product will result in a surrogate measure of how much of the organism was present initially in the specimen. I guess moving on to the host now, host genetics are important to disease presentation. There are many examples of immunodeficiencies where the host has deficiencies in the innate immune system, the humoral immune system, or the cell-mediated immune system, resulting in infections. The host genetics more broadly can also be involved in different responses to illnesses. and essentially we're looking at all range of illnesses and host variability in the way the host processes those illnesses. If we look at infection, the trinity of infection involves the host and the host immune response, and it involves the pathogen. But the third important component in this is the inoculum. So the amount of organism present, the type of host, and the type of pathogen we're dealing with. And this triangle is very important and is worth remembering, because analysing any infectious disease without analysing these three components, you're only then looking at isolated factors. How do we measure the host response? Well, we're very familiar with this in clinical medicine, and we in fact do this all the time. We're very familiar with measuring the full blood count, the neutrophil count, whether there's a left shift in the neutrophils, whether we have a raised CRP or a raised procalcitonin. These are all established measures of how the host is dealing with an invading pathogen. There are a range of newer biomarkers and artificial intelligence putting different biomarkers together in order to determine exactly what's happening at the host level. When we deal with infection, it's different to all other specialities in medicine in that we're dealing with two organisms. We are dealing with the host, and we're also dealing with the invading pathogen, whether it be a virus, a fungi, a mycobacteria, or a parasite. The genetic determinants of both the host and the pathogen play an important role in determining the disease presentation. So what do we look to find out about the pathogen and the genomics of the pathogen? Firstly, we're interested in whether an organism is present or absent. Secondly, we're interested in the amount. We're interested in resistance to antimicrobial organisms, whether the organism is viable or not, and the clonality of the organism if we're dealing with an outbreak. Many of these can be determined through molecular mechanisms. So I'd ask you to think now about the question that's being posed in infectious diseases. Are we posing a narrow question such as, is the pathogen present in the specimen? Or are we posing a broader question, is there any bacteria, fungi or viral pathogenesis present? There's a few confounders which are typical of confounders in microbiology. Firstly, sterile versus non-sterile sites. So blood cultures and CSF, classic sterile specimens, much easier to work with in the laboratory because we are determining normality as being the absence of organism. So any organism potentially present in those specimens could be significant. In wound swabs, urine, faeces, non-sterile specimens, we have a much greater difficulty in determining the significance of organism. Often the respiratory tract, although we think it's sterile, is not sterile, and determining the amount of organisms present in that site can help us determine whether the organisms are significant or not. If we want to determine viability of organisms, we still don't have a molecular mechanism for doing that. We really need culture in order to determine viability of organism. So narrow question nucleic acid amplification technique. There's a variety of techniques, and I just want to run through some of the available platforms to answer these questions. There are platforms that look at a single pathogen. Is that pathogen present or absent? And our classic example here is the COVID epidemic, SARS-CoV. In the last couple of years, we've been doing huge amounts of molecular work identifying SARS-CoV-2. that is done on a range of platforms that have essentially cycle specimen, detect the organism, and then report out the number of cycles present in order to detect that organism, which is what we call the CT value, and that gives us some degree of insight as to how much of the SARS-CoV-2 virus is present in that specimen. We also have duplex platforms that have been set up to detect two significant pathogens in a specimen, and a classic example here would be flu and RSV. So in a normal winter before COVID, we're really dealing with flu and RSV, and it's really a question of one present, two present, or none present, and that is a very cost-efficient way of doing a test in a winter viral outbreak where it's likely to be either flu or RSV. In addition to that, in recent years, we have multiplex testing. Multiplex testing combines many PCRs into the one reaction so that you can look for a range of organisms in a specimen, often organisms that are likely to produce a clinical syndrome, and this is what we call syndromic testing. There are many different platforms to do this, and I'll just run you through some examples. Many of these can be used to detect SARS-CoV-2. This is the Kefid GeneXpert, which has been commonly used during the COVID outbreak, and generally will detect 1 up to 4 pathogens depending on the cartridge. But for SARS-CoV-2, a single monoplex assay. These assays are put in modules, and depending on the size of the laboratory, there's a number of different drawers that can be used to insert the cartridge. So here we see a cartridge being inserted into a GeneXpert machine. This one is actually a tuberculosis cartridge, so what we're dealing with is MTB and rifampicin resistance. So this is a duplex assay, and it's very widely used throughout the developing world, and it enables both detection of MTB and also detection of rifampicin resistance. So a highly useful duplex assay that can be done in rapid time to detect that pathogen, and also illustrates the next point, which is that PCRs can be used in this sense to detect genes that are associated with resistance. So particularly rifampicin resistance in TB, but also genes like the MEK gene in Staph aureus. We commonly use these duplex assays to detect the presence of MRSA as well as the presence of Staph aureus. So moving on to other forms of PCR, we can ask a much broader question, which is anything present in the specimen? Anything from the bacterial kingdom or from the fungal kingdom? And we do have in-house PCRs which are structured around looking for the bacterial 16S PCR, which is a conserved sequence in bacteria, or the 18S PCR in fungi, which is a conserved sequence in fungi. And then we can move on to talking a little bit about metagenomics, where everything in the specimen is amplified, and then there's a bioinformatics exercise of putting together large amounts of data. So this is an example of a multiplex PCR. This is the bioMérieux BioFire panels, and these are also done rapidly and quite simply. So a lot of the process of the PCR is internalized. The specimen is added, and then automatic extraction, amplification and product detection occurs. It does enable a large amount of organisms to be looked for simultaneously. This panel down here shows that there's a range of bacterial and viral pathogens in CSF, and in this case there's 14 pathogens in CSF that can be looked for simultaneously. So this is a really useful technique when you have a patient that you've done a lumbar puncture on and you want to ask the question, are all of the common pathogens present or absent in this CSF specimen? This will take approximately an hour and is revolutionising the way we approach diagnosis of infectious diseases in critical specimens. So as well as CSF, there are similar panels for respiratory pathogens, and if we look at the pneumonia panel, there is a range of organisms that may be present in, say, a ventilator-associated pneumonia, but also other organisms that may cause community-acquired pneumonia. This panel also has the ability to detect the amount of each organism that's present and can quantify these organisms, as well as, interestingly, identifying resistance genes that may be present. So once again, in an hour you can get a vast amount of information about organisms from a non-sterile site, a deep respiratory specimen, either a sputum or a bronchoalveolar lavage. And this is still a tool we are learning how to translate that into clinical medicine, but the amount of information that can be achieved in a short period of time is really quite significant. In addition to that, there are similar panels that are used for blood culture identification. So when a blood culture flags positive in the laboratory, a blood culture panel can be used to identify multiple organisms, but also resistance factors. The resistance factors are up the top there, so KPC, MEK-A, VAN-A, VAN-B, all resistance genes for organisms that enable us to make good clinical decisions about what antibiotics are necessary. That's just a list of the meningitis encephalitis panel, which lists a range of viral pathogens, CMV, herpes, human parechovirus, very important in children, varicella zoster virus, as well as bacteria, some of which are present in neonates and children, so useful in that context, as well as strep pneumoniae and cryptococcal PCR. So multiplexes can include viruses, bacteria and fungi, and once again a powerful technique. I just want to, before finishing, move on to just touching on whole genome sequencing and the usefulness of genome sequencing. So many of the different techniques that are available for whole genome sequencing use a series of reads of varying lengths, and then construction using bioinformatics of those reads into a whole genome. Often there's a reference genome, and if there is a reference genome, the process of assembling the whole genome will be much easier. There's other techniques that do longer reads, such as the Oxford nanopore, and result in, with whole genome sequencing, it results in the generation of, essentially, of a family tree of organisms, so that organism can be compared to other organisms. Now, where we have been using this in particular in recent times is also in the SARS-CoV-2 pandemic. And what that enables us to do is to see mutations in the virus, and we know that RNA viruses mutate very frequently, and then we can look at the distance in relative terms between a known organism and a new organism in order to determine the evolution of that virus and how that's occurred. And that can have really major impacts in terms of identifying clusters of organisms, transmission and epidemiology as we watch the spread of that. The Oxford nanopore is a very small machine, and it really is Star Trek-like in its ability. But in general terms, whole genome sequencing uses a series of steps. So genomic DNA is extracted, it's then fragmented, amplified, and sequenced. Sorry, not amplified, it is sequenced, and then the data is analysed in a bioinformatic way. So that's high-level maths, large data, a lot of computing power. So we use this epidemiologically, and I've mentioned the SARS-CoV-2 example, but additional to that, we can look at common organisms throughout the hospital. We've been looking at Serratia marcescens spreading in wards, and it enables you to relate organisms together, and if you see the same organism spreading in a unit, then you can track an outbreak of multi-resistant organism or of antibiotic selected organism within an ICU unit, for example. In addition to that, organisms often have plasmids, which are self-contained circular DNA within the genome, and it is possible that a plasmid can also cause an outbreak in different organisms. And by sequencing the plasmid and looking for connectivity between organisms, we can detect what are being called lately plasmid outbreaks. So this is very important for infection control. Lastly, I want to just talk a little bit about metagenomics. Metagenomics enables a specimen to be analysed to look at everything that's present in that specimen. And this enables the detection of novel pathogens. So the example I'm using here is an encephalitic patient with an abnormal CSF, but no organisms detected that we know of, can be meta-sequenced in order to reveal sequences suggesting a new pathogen. And in our hands, we were able to do this to identify a PEGI virus encephalitis that would have otherwise been an undiagnosed encephalitis. So we sequence everything in the sample and then construct a map of the microbiome in that specimen. This has also been done in the research field, particularly looking at the gut microbiome and looking at all of the organisms that are present in the gut and associating different organisms in a microbiome with different clinical states. As well as the gut, it's been done in selected regions like the mouth and the vagina, and this leads to the terminology of mouth and vaginal microbiome, describing the entire organism complement of those sites. So very useful technique for looking at large changes in organisms in non-sterile sites in particular. And I think we'll hear more about this as we go further on. So that concludes my talk. Thank you very much for listening today, and if we were a live audience, I would ask if there are any questions, but I appreciate your listening. Thank you very much.